Imagine an animal who has the ability to regenerate two complete organisms when its body is severed in half. Or if you sever it into 10 pieces, it will regenerate 10 complete versions of its original self: head, eyes, digestive system, and all. And amazingly, you can cut up this animal into 279 pieces, and within a few weeks, each bit will have regenerated a full new body. That animal is an amazing little flatworm known as Planaria, a favorite model organism of scientists studying regeneration. Scientists like Dr. Michael Levin, who are revealing previously unknown mechanisms of control that function outside of the DNA-centric view of cell fate, a mechanism which harnesses the power of bioelectricity. Now, before we jump into my conversation with Dr. Levin, let's set the stage with some foundational knowledge. Now, you're probably familiar with the idea that DNA is the software of life, dictating the proteins that make up the hardware of every cell. But here's the twist. Dr. Levin's research reveals a powerful layer of control beyond genetics. He studies bioelectricity, the voltage gradients and electrical signals that cells use to communicate, just like neurons in the brain, but all throughout the body. These signals serve as a kind of software running on top of the protein hardware, sort of like an API, coordinating processes like regeneration and shaping how organisms develop from a single cell into a complicated symphony of specialized cells. And just like all other software, the bioelectrum has the ability to be hacked and reprogrammed by the user, in this case, Dr. Levin himself. He and his team have been able to tweak these bioelectric circuits to produce two-headed worms. And the amazing thing is that when these two-headed worms are once again severed in half, they regenerate not into normal one-headed worms, but into worms with two heads and with no changes to their genome. These bioelectric circuits exhibit a sort of memory and ability to problem solve, which has great implications on our understanding of intelligence and will undoubtedly impact the future of AI, neural nets, and machine learning. Now, what if this ability could be unlocked in humans? What if we could reprogram cells to repair damaged tissue, reverse birth defects, or even combat cancer, all by modulating these electrical signals? In today's episode, we'll explore how Dr. Levin and his team are rewriting the rules of biology, from decoding the bioelectric code to regenerating frog limbs and developing bioelectric therapeutics. We'll unpack the science, the potential applications, and the profound implications of this exciting field. Dr. Levin is a distinguished professor at Tufts University, where he leads the Allen Discovery Center and the Tufts Center for Regenerative and Developmental Biology. He is a once-in-a-generation scientist pioneering a developing field that's bridging the space between biology, computer science, and even philosophy. Join me in Vivo with Dr. Michael Levin on decoding the secrets of regeneration. So, Dr. Levin, I think had I known how important ion channels would be to the future of biology, I think I would have paid attention a little bit closer in cell biology class all those years ago. And what I also didn't appreciate or think that I would see in my lifetime is somebody coming up and sort of challenging the DNA-centric model of biology that we all really understand and appreciate, that's printed in every single textbook today. And that's basically that DNA is the arbiter of cell fate. It controls the whole protein inventory of the cell and runs as a sort of software. That's that analogy that we always hear. But you have a very unique perspective based on your work in Planaria, amongst other organisms. But I think in order to understand your thesis, we should start with Planaria. What is Planaria, and why is it such a fascinating organism?
Yeah, maybe if we could just take a quick step back, and
Huh?
I just want to say one thing about what you just said. So, actually, there have been a whole bunch of people in general arguing back and forth around the centrality of DNA and all of that, right? Most recently Dennis Noble and many people before him. So I'm certainly not the only one, but what we do need to think about, I think, is what do we really mean by this? In other words, when you say that a particular set of mechanisms or a particular level of description or whatever is that's the explanatory level, what do you really mean by that? And I just want to talk about that for a quick second,
Absolutely.
because we're going to get into a bunch of other examples from our work and all of that. And I want to be clear that I'm not saying that genetics and genetic information isn't important. But I do have a specific criterion for what we mean by when we say these things are central. And what I mean by that is that we want formal models that facilitate discovery and facilitate invention. And this is important because I'm gonna describe to you a number of experiments, and everything that I talk about, one could look at that and say, oh, I think I can explain this in terms of genetics and biochemistry.
Okay.
And this is true because, much like with every act of human behavior and every physical event, if you wanted to drill down to the lowest level and look at the kind of the low level hardware that made it happen, yes, you will always find the chemistry and whatever quantum physics below that.
Right,
It's,
right.
it's not you don't find magic, you don't find anything like that. My claim is not that when you look underneath these examples I'm going to tell you, you're going to find something other than the conventional laws of physics and chemistry. My point is that if you do start out with that as your primary level of description, there are a whole variety of discoveries, including some very useful things for regenerative medicine, for bioengineering, that they are going to keep you from finding. And
Right.
so I just want to get that clear, is that looking backwards and providing reductive mechanistic explanations for things is always possible going backwards. But what we want to understand is what kind of worldview helps you discover these things, and what kind of worldview actually predicts some of these things as consequences.
Right, and,
Right.
and and I hope to get into this a little bit later. It's also a product, I think, about perspective and where you're coming from. As a
Yep.
molecular biologist, I dealt with a lot. Dealt a lot with DNA transfections and that sort of thing.
Mm-hmm.
And you come
Hmm.
from a computer
Yeah.
science background, and so I think that has a lot to do with the way you approach the problem. We'll get into that later, but I think that's a little bit of
Okay.
what you're getting at as well.
Yeah. Yeah. Okay. Yeah. Let's talk about planaria. So planaria are an amazing creature. They're a pretty popular system nowadays for regeneration and for behavioral pharmacology and for some other things. They are a free living flatworm. There's many species. It's a group of free living flatworms. They are anywhere from half a centimeter to a couple of centimeters in size. You can obviously see them with your naked eye. They are not like earthworms in that they are true bilaterians. So they have bilateral symmetry. They have a true
Yeah.
brain. They have many of the same neurotransmitters that we do, and so they are similar to a kind of an ancestor of our lineage. So they have some interesting properties. The first is that if you cut them into pieces, every piece will give rise to a perfect little worm. So they have this amazing like holographic kind of property where
Right.
they regenerate. They regenerate every part of their body. They can regenerate, and almost every part of their body can regenerate. There's a couple of small exceptions.
Right, and I think
Uh,
just the term holographic, I think sometimes gets misconstrued with some woo stuff. What you mean by that, I think, is that every cell in the body has the information coded in it to make every other part of the body.
Correct. I mean, we'll talk about information and how you code information and all that, but functionally, what I mean is exactly this. I mean, holography is it's an area of engineering. It isn't a woo thing. It's what it means is that once what you have a material where you can cut it into pieces and you can recover the whole information.
Right.
You don't end up you don't end up recovering just the piece that you cut. Right.
Correct.
So that's all I mean by that.
Yep.
Um, so yeah, so they have that regenerability. Also, they're smart. They can learn, so they can learn in a variety of training assays, which means that you can do behavior and brain regeneration in the same animal. That's very unique. That
Mhm.
means you can train them on something, you can cut their head off, they regenerate a brand new brain, and then you can, for example, find out that they remember the original information.
Mm.
So that's pretty amazing, and that's
Mm.
not easily doable in most other systems, although there have been attempts. Um, they are cancer resistant, so they have very, very low rates of cancer. They survive carcinogens and all of that. And the asexually reproducing ones, which basically the way they reproduce is they tear themselves in half, and each half regenerates. Right, the head makes a tail, and the tail makes a head. Those lines of worms are immortal. They don't age. So there's no such thing apparently as an old asexual planarian. So that's that. Those are some amazing things. One implication of the fact that they reproduce asexually is that, unlike us, who go through a sperm and egg stage, so that if you get a mutation in your body, your children do not inherit that mutation. In planaria, that's not true. So any mutation doesn't kill the stem cell, gets amplified into the next generation because that cell has to rebuild, right, a bunch
Right. Right. Right.
of body tissues. So isn't that wild? I mean, we just to start with that right off the bat, what in your biology education would have led you to predict that the animal with the noisiest genome that accumulates for 400 million years, has been accumulating these somatic mutations, is actually the one that's highly regenerative, cancer resistant, no aging, right? Isn't that sort of opposite to everything that we've been told about the role of the genome and how all this works?
Yeah, absolutely. And something that unless you're deep in the world of sort of model organisms and studying, you don't often hear of Planaria, period.
Right.
And so, yeah, fascinating creature. Absolutely.
Yeah, yeah, and they also another thing I forgot to mention. They are which finally after years, this puzzled me for many years, and finally I
Mm.
think we understand what's going on. Uh, they also are not only resistant to transgenesis, so you can make transgenic flies, mice, whatever. There are no transgenic planaria, nor are there any planarian strains with abnormal morphology. So you can call the stock center and you can get flies with curly wings and
Hmm.
mice with
Hmm.
weird tails and whatnot. Yeah, the only abnormal strain of planaria that you can get is our two-headed form. It's not genetic. And so there's actually something very interesting going on here, which we can talk about if you want, which is why why is the genetic level not amenable to in planaria, not amenable to that kind of manipulation?
That's fascinating. The transgenic point there, I had never
Mm,
heard. That is something.
yeah.
What are your thoughts on that? Because back to the DNA-centric view of biology, that's something you'd never imagine.
Yeah. Well, so the way we make our two-headed worms is by changing the information in the bioelectric circuit that they have. So they have a stable bioelectric pattern within their tissues that says one head, one tail, and that pattern is a kind of memory, literally, because you can alter it with a brief physiological experience. You can change it. You can say no, two heads, and from then on, all the pieces regenerate as two-headed in perpetuity. So the DNA has not been changed, and yet the pieces will continue to regenerate as two-headed. So that tells you right there that the answer to how many heads does a planarian have is not in any sense coded in the genome. What the genome does do is produce some hardware that by default self-organizes a bioelectrical pattern that says one head, in the same way that when you buy a calculator from the store, you turn it on by default, the first thing you see is zero, reliably 100% of the time. That's what it does. So yeah, so you make some hardware where the circuit is such that under default conditions it says one head. But like a reprogrammable kind of device, which I think these are, you can actually you can change that information. And so what I think is going on in this case is that and we've now done a bunch of computer simulations of this kind of process. It goes back to asking the question, how does evolution proceed with a very competent substrate? Most of the time when we think of evolution, when people model evolution, they go straight from here's your genotype. Your genotype determines the phenotype, and the mutation operator works on the genes, the selection works on the phenotype, and you put it in a cycle, and then we all know how that turns out. And people do genetic algorithms and things like that. What you're missing is the middle layer, which is morphogenesis. It's and the reason it's important is that morphogenesis is not a passive mapping from genotype to phenotype. It's a problem-solving intelligence,
Hmm.
and I mean that quite literally. It's a system that is able to solve various problems. And what happens there is that just imagine for a second, and we'll get back to planaria. Um, we showed a while back that if you have a tadpole, tadpoles have to become frogs and they
Yep.
rearrange their face. And what we showed is that if you scramble the craniofacial organs of a tadpole, you still get a normal frog. They will actually go through novel moves, novel movements, and eventually you get a normal frog. Right.
Problem solving.
So
Yeah.
just imagine. Yeah, yeah, right. So navigating the anatomical morphospace. You have an anatomical space, and you're navigating from where you are now to where you need to be. And then we'll get back to how you know where you need to be, which is these bioelectric pattern memories. But getting there is not always easy because you may get taken off course. There might be a barrier. You might be
Hmm.
broken in various ways. There's all kinds of things you might need to deal with. So just imagine. So you're a tadpole and you suddenly have a mutation, and most mutations do more than one thing. So you end up with, your mouth is off to the side of your head, but also something else positive is happening in your tail somewhere, right? Under the standard model where the genetics determines the morphology directly, that tadpole is dead. Your mouth is off to the side. You can't eat. That's it. And so now evolution can't explore the positive benefits of that of the other positive benefits of that mutation. You have to wait until somehow whatever that feature is appears by itself without the thing that moves the mouth. That's very difficult in a pleiotropic model like this, where every gene affects multiple different things, and evolution should take a really long time. Instead, what happens is the mouth will fix itself. Okay, the mouth will actually move to where it needs to be, and then by the time this organism comes up for selection, that part's fine, and now you get to explore whatever else is going on with this mutation. So what's happening is that when the material over which evolution is operating is not passive, the way that we engineer with wood and metal, but it's not even active matter. It's actually I call it an agential material,
Right.
because it has certain autonomous abilities to get certain things done despite novel circumstances. What happens is that it hides information from selection. When selection sees that tadpole, it can't tell that, hey, actually your genome wasn't very good. We need to weed you out, because selection just sees the phenotype. And so by having these self-repairing properties and this ability to get to where you need to go, and we have lots of wild examples of systems that do that, you end up hiding genomic information from evolution. And when you do that, evolution has a really hard time, selection has a really hard time picking the best genomes. So it has a hard time seeing the structural genomes. Where does all the work get done? Well, all the work gets done on improving the competency. So instead of improving the structural genome, what you end up doing is improving the mechanisms that allow you to get the job done, even if your hardware is a little wonky. But that starts a positive feedback loop, because the minute you do that, you start generating even more competent tissues, which hide even more information from selection. And so you can see this in the simulations that we've done. Evolution running on this kind of on this kind of agential material acts completely differently. What happens is the pressure on the hardware flattens out,
Right.
and then all the work gets done on the algorithm itself. So now just imagine you end up, and I don't know why certain lineages stop at different positions along the spectrum, but you've got a spectrum. On the left side of the spectrum, you've got something like C. elegans, which is incredibly hardwired, right? So every cell is numbered. Every C. elegans has exactly the same number of cells. They're all lineage determined. As far as I know, it doesn't regenerate much of anything. That's it. Then on the opposite end, you have Planaria. In Planaria, that ratchet went all the way forward, because the material is so unreliable, because you're going to be accumulating mutations, right? Because of this crazy kind of somatic inheritance that they practice, the genome, the genome's going to be noisy. So all the work went into making an algorithm that, for all intents and purposes, ignores a lot of the information in the genome. This is why transgenesis doesn't work. This is why we can't make mutant lines in Planaria.
Wow.
For the same reason that the natural mutations that have been accumulating for hundreds of million years don't change. All the work has gone into that algorithm. And in between, you have things like salamanders, and then things like mammals that are sort of, they can make up for some things, but not others.
Interesting. So it's as though it's as though this defense mechanism, for lack of a better term, evolved over time to protect this noisy genome, and that's why it's and that's why it's pushed all the way to the other side of the spectrum. That's fascinating.
I think that's true, and I think that in this particular case, that's why it's gone so far. But I also think that fundamentally, this issue of and this is really interesting, this issue of working with an unreliable substrate is true for all life from the very beginning. This is one of the ways in which computer. So I use a lot of computer analogies, both in my talk and in my my discussions and in the research. But one way in which the current computational paradigm absolutely does not match biology is that in computer science, what you try to do is separate the layers. You try to encapsulate the layers, so that when you're writing code at the high level, you don't need to worry about, well, geez, if the copper gets a little warm, all my registers are going to float off, and the numbers will start to. You don't worry about that. You assume that the hardware is going to do what it needs to do, and you're operating at a particular level. Biology, I think, commits from the start to an unreliable substrate. You never know what you actually have. You're guaranteed to mutate over time. You don't know what never mind the environment, but you don't even know. And again, there's many amazing examples of plasticity in biology, where wrong copy number of your genome, it's fine. The wrong number of cells, also fine. Wrong cell size, no problem. They figured it out, because even normal embryos under normal circumstances, we're not assuming that everything, except maybe C. elegans and things like it, we're not assuming that everything was going to go correctly. They are problem solving in real time every single time, and that's what accounts for the incredible plasticity. And in some cases, like Planaria, just it like takes off exponentially.
That's fascinating. Do you know, is there a correlation between genetic or genome noisiness and this ability to regenerate? Is that something set in stone?
It's a good question. I don't know if a good study across phylogeny has been done. I'm not sure. I mean, probably. Yeah, I mean, it's probably information about, for example, axolotl genomes and things like that. Yeah, that could be done. I'm not sure. I'm not sure that's been done.
And then sitting here as someone who did a lot of tissue culture work, you're talking about
Mm.
transgenic making a transgenic planaria is virtually impossible. Can you ectopically express genes in not even ectopically for a moment?
Doesn't seem. I mean, it's entirely possible that somebody will crack this. People have been
Right.
working really hard for 40 years on this now, and nobody's really done it. Maybe somebody will crack. I mean, presumably there's a way to overcome it, right?
Right.
There's got to be, in terms of synbio and whatnot, there's got to be some way to overcome it. But the natural situation is very interesting. It's just not taking that information as literally as other systems do.
That's fascinating.
And yeah.
Now there's a beautiful sort of story or analogy I've heard you told. We teach biology students that DNA is the software, the rest is the hardware, and you like to refer to the bioelectric circuits that you're talking about as more of the software running on the hardware, which are the proteins, and that there's a similar perspective change happening where in the early 40s you had computer engineers. When you wanted to manipulate a computer, you'd move around wires and physical gates and that sort of thing, but later we moved towards software, and right now in biology there's this sort of shift towards being able to manipulate these bioelectric circuits the way you would software. So can you tell us a little bit about those bioelectric circuits and how and why you sort of look at it that way in terms of software or an API even?
Yeah. Well, the first thing to say is that I don't believe there's any one right answer to any of this. Josh Bongard and I work on this polycomputing framework where
Mhm.
basically what we're looking at is the ability of different agents for different reasons to come up with different models and different ways to hack one particular some particular physical system.
Right.
And I think all of these metaphors, and I think they're all metaphors. So pathways, none of these things are real, right? So
Right.
so
Right.
so
Right.
pathways, all that stuff, including memory and circuits and all of it are different metaphors. The uh, reason to have any of these metaphors is that they facilitate certain kinds of interactions and they prevent you from having other kinds of interactions. So it's fine if you want to say that the genome is the software. That's great if that enables you to do something interesting. So
Mhm.
in all of these cases, these metaphors allow you to take tools from some kind of field of study, for example, computer science, so something like that, and apply them to in a particular case. So what does that allow you to do? I prefer, in most of our work, I use a different metaphor, um, which is that what the genome actually sets is the proteins that every cell gets to play with. It is literally encoding the physical hardware of life. Now, once you have those proteins, you end up with several different layers of software. I happen to focus on bioelectrics, but of course, biochemical
Mhm.
information is very important. And we have studied the way in which, for example, gene regulatory networks can learn and have memory and those kinds of interesting things. And other people study biomechanical kinds of layers that also exist. But we study bioelectrics. So what happens is the easiest way to think about bioelectricity is to kind of make two mental shifts. What you do is you start with neuroscience and
Yeah.
you think, okay, here's the story of neuroscience. So in neuroscience, we are an emergent collective intelligence. We are made of a whole bunch of neurons. We have goals and memories and problem solving abilities in three-dimensional space and also in some other weird spaces like linguistic space and
Mhm.
chess
Mhm.
world and so on. So we have the ability to navigate these spaces because the cells of which we are made are connected by an amazing system. I call that class of systems cognitive glue. They are the cognitive glue that operates in our brain is bioelectricity. So the neurons form networks. These networks perform computations. They do some other things, and together that enables us, while the neurons are working in their local physiological and metabolic spaces, the collective of which they are part, those giant networks, end up projecting their intelligence into other spaces. Okay, so that's roughly the story of neuroscience. So now all you have to do to understand developmental bioelectricity is this. First of all, to realize that all of the mechanisms that are being used to do all this are extremely ancient. So ion channels, electrical synapses, neurotransmitters, all of these things are very ancient. So that raises the very obvious question: what were the networks thinking about before nerve and muscle evolved, and before we could run around in three-dimensional space and solve classic behavioral problems? What were the cells doing? These same networks existed. What are the precursors of the brain and the neurons? So that's the first thing. And the second thing is just to say that if you're willing to look at different time scales, so neurons are very sped up. They work at the millisecond time scale. The bioelectrics of the rest of the body is hours, let's say, that kind of thing. And if you're willing to think about projecting intelligence into other spaces that are hard for us to visualize, for example, anatomical space. So navigating anatomical space while, and that not just mechanically rolling along some rules and producing something, but actually being able to get there even if circumstances change. Right, and there's an incredible amount of that kind of plasticity in development. That's William James's definition of intelligence: the same goal by different means.
Right.
Okay, same ends by different means. So if you're willing to change the time scale and the problem space, everything works the same way. In fact, I used to have my students do this manually, and then we with some colleagues, we produced an AI that does it for you. You can take any neuroscience paper, and you can just do a find replace, and every time it says neuron, you just replace that with cell. And every time it says millisecond, you say hour, and now you've got yourself a developmental biology paper. because I think literally, right, that's what evolution has done. Is it's pivoted some of the exact same tricks through different spaces. And so what happens is that let's tell the same story developmentally. You've got a bunch of cells, not neurons, just early embryonic cells. They're all expressing ion channels. These ion channels produce voltage gradients. They are sitting next to each other, connected in some cases with electrical synapses known as gap junctions. So that allows the information to propagate or not, because both the channels and the gap junctions are themselves voltage sensitive. So that gives you these amazing feedback loops that allow you to have memory and historicity and things like that. And the information that propagates through this network does exactly what bioelectrics did in bacterial biofilms, which is integrate information across space and time. It's a cognitive glue that binds those cells together into common purpose. What is their common purpose? It's to together navigate, instead of three-dimensional space, which is what your neurons are doing, it navigates anatomical morphospace. You start life as, let's say, a single cell, and then you have to get to the region of morphospace that corresponds to a normal, let's say, human body or something. So these electric circuits that I'm talking about are simply collections of cells that have ion channels in them. It's an excitable medium, like your retina or parts of your brain, and they can hold spatial patterns of electrical information that we try to read out, the same way that the people who try to do neural decoding try to read brains, right, and decode that information and infer what is the animal thinking about, what memories does it have. That you can do exactly the same thing in the body, but what you're going to pull out are memories of pattern. You're going to be able to see what this being is trying to, where in morphospace is it going? What does it want to do? The way that the neuroscientists are trying to predict animal behavior from reading their brains, you can do the same thing in the rest of the body, but what you're getting is anatomical information, not 3D motion.
That's absolutely fascinating. I like the way you use the idea of glue. That's a very good analogy. And if we were to look at these bioelectric circuits as a sort of useful software, you have to have some ability to do something useful. An example of what you've done is with the Planaria, right? You've cut them in half, and you're able to treat them in such a way where you change the bioelectric circuits to develop a second head where there should be a tail. Can you explain how that's actually done practically in the lab? Is it by application of actual currents, or are you changing sort of the salt concentrations and ion concentrations in media?
Yeah, right. And so we can. So at this point, we've shown control of head number, but also head shape. One of the cool things is that we can get them to make head shapes that belong to other species of Planaria. So 150 million years evolutionary distance, same hardware, different head shape, same genome.
Wow.
by right yeah so how do we practically do this? No, we do not do electric field application or anything like that, although people have in the 40s. Marsh and Beams published some of this using electric current. Yeah, we don't use electric current because it's kind of a blunt tool, and we have now a lot of methods that have been developed in by neuroscientists. And we take everything from neuroscience, like everything works the same way. So in other systems we used optogenetics and and pharmacology and so on. Changing the ion concentration usually by itself doesn't work because there's a lot of stability in these circuits, and so they are really good at doing what they need to do, even if the external environment changes. So by itself, that's hard, although sometimes you can. What we end up doing is targeting the ion channels that are producing the bioelectric states. So that means either pharmacology, so drugs that open or close these channels, or RNAi. Or RNAi, you can knock down some of these channels in some cases. Yeah, the idea is to control the gap junctions and the ion channels.
Understood. Uh, I've got a question that I'm not even sure I know how to ask, and it's part of your thesis is that not just yours but others, the bioelectric circuits are sort of helping to coordinate the movement in anatomical space during development and also during regeneration. Where is that? Because there's an element of memory involved here, like you said,
Oh, yeah.
the ability to store these pattern circuits or patterns
Yeah.
in these circuits. Where is that information, the base level information, coded? Is it coded in the genome? Or, from day one, when you have one cell that becomes 2, 4, 8, where are these gap junctions pulling this information from?
Yeah. Okay. So I'll tell you the kind of mechanistic answer, and then we can talk about the deeper philosophical issue. So the deeper the mechanistic answer is that if you have a particular electrical circuit, it depending on the circuit, it can have the ability to self-organize, spontaneous symmetry breaking, and then self-organization of patterns. So in biochemistry, the example of that is Turing patterns, right? So as Turing and many people since then showed, you can have a completely well mixed set of chemicals, and under specific conditions, patterns will self-organize in that excitable medium, and you get things like zebra stripes and leopard spots and so on. Okay, so something similar is happening here in the sense that you can have a completely homogeneous field of cells, and electric voltage patterns will self-organize. And if you, in the case of the frog embryo, for example, where you have a known starting point, which is the fertilization, you can watch the channels come. The channels are maternal. They come to the surface. They get redistributed, and then you can watch the bioelectric patterns self-organize. Okay, so that's the mechanistic answer. Now it's interesting to talk about kind of the philosophical meaning of where is information. This is a very subtle question. We are used to easy scenarios where some person writes down some information or writes some, encode something, and we can say, well, there it is, because that's where we encode it.
Right.
This is different. In the case so for example, in the case of the Turing pattern, when you get curlicues of a particular width, you can say, so where was that width recorded? Well, it wasn't written down. It's not written on top of the little molecules, and it wasn't really directly anywhere that you put in. Another example that we can think of is a fractal, and I can, if you want, for the show notes or something, I can send you a couple pictures of it. It, like, if you do a Halley plot of a formula like z squared plus 7, you get this incredibly rich, complex, biological looking thing with all kinds of patterns. Where is that pattern? You don't really want to say that pattern is in the formula, because the formula is only 6 characters, and that's a degree of compression that is unthinkable. So where is it? Well, it's wherever the laws of mathematics live. It's in some sense what evolution is doing, and I feel strongly that in biology we're very used to 2 sources of information. There's genetics, and then there's environment. Okay, and I think we're missing a big third one, which is what mathematicians study. Many mathematicians think that we don't invent mathematics, we discover it.
Right.
And what that means is that there's a world of truths, the distribution of prime numbers, various true theorems of number theory, and facts about triangles and whatnot, and that all of these things are true, no matter what happens in the physical world. In other words, you could have shuffled all the constants around the Big Bang and these things would still exist.
Right.
So there's some independent world of mathematical truths. I think biologists need to take this very seriously. And I think what evolution is doing actually is searching that space. I think when evolution produces physical embodiments like embryos and things like that, what it's really making is pointers into that latent space of possibilities. And I think we need to get over, like right now, there's a tendency which I think is, um, it's very much like it papers over a real Mysterian kind of approach to things, where we find these things that are kind of emergent like that, and then people say, well, that's just something that holds. It's just a fact that holds. So when Stu Kaufman finds these amazing facts about gene regulatory networks, or something, the way that all like all the stuff in uh, um, Darcy Thompson's book on growth and form, and all these like amazing things that are not directly encoded by either the environment or the genome, then people will say, well, it's just a fact that holds about this. It's just something that holds. And I feel strongly that we should not assume that there's a random bag of things that hold, and we should just wait until we're surprised by them and then sort of catalog them, and that's that. We should have a principled research program, much like the mathematicians do, about asking what is that space of affordances. I think the null assumption should be it's a structured space. Like all of science, you don't assume that the world is chaos. You assume that there's order underneath, and that we're going to, until proven otherwise, we're going to systematically explore this. And so I think that's what we should take seriously. And I think we should be exploring this space, and that's why I see some of the things that we make, like xenobots and anthrobots and so on, specifically with normal genomes, but very unusual and novel forms and behaviors, which were never selected for by evolution, which live in exactly the same environment as normal embryos do, and yet they have these novel forms and behaviors. Where do they come from? They don't come from selection. So that's why. I'll stop there to say that the question of where does it come from is a subtle question, and I think we need to really shift our thinking from the usual types of answers, where either it's written right there in the DNA, or it's just the thing that holds about the world.
Right. Something that's ultimately a space, like you mentioned, that's being tapped into, and all the possibilities are being drawn out of it. Yeah, that's fascinating. And you just mentioned xenobots, and your work with that is also fascinating. And while we're sort of on the topic, your sort of thesis here on the bioelectron and bioelectricity has some implications on intelligence, and I really love the way you approach this idea. I mean, intelligence, in some sense, in a very basic sense, is being able to solve problems and to code and recall information. And it seems as though that the cells that we're talking about here, with the gap junctions and all, have that ability. And so, what is your definition, or how do you view intelligence in light of your research?
Yeah, just to be clear, again, I'm not suggesting that my definition is the only one, or the correct one in any kind of unique sense. My definition for sure doesn't capture important aspects like play, emotion.
Right.
There's some things that are not captured here, and that's kind of on purpose. I want to focus on parts that are experimentally tractable.
Mhm.
So I'm not claiming that this covers everything, but this is a nice, tractable, useful definition. And I follow William James's definition, which really does focus on problem solving. And it says his definition is same ends by different means. So what it focuses on, I like it a lot because it's very cybernetic. It doesn't say you have to be a brain, and it doesn't say you have to be evolved versus engineered. It's very generic, right? It doesn't say what space you're working in. It doesn't confine you to some specific kind of problem that you're solving. It's very general, and what it focuses on is two things: the ability to have goals. And so this is a part of my cognitive light cone model, and the idea that all active agents, no matter what their composition or origin story, can be placed on the same diagram by virtue of the size of the largest goal that they can possibly pursue. Okay, so goal directed systems, that's one key thing, and the other key thing is having some degree of competency to achieve that goal even when things go wrong or change. So that could be very little, that could be quite a lot. And I talk about, for example, like there's a spectrum you can imagine, like two magnets, right? Coming together. You put a piece of wood between them. So the magnet is never going to come around and get to the other magnet, because to do so they would have to temporarily get further from their goal, and they're too stupid
Right.
to do that. They can't go against the gradient, so they'll just stand there like this. But that kind of delayed gratification, some animals can do it, some cells can do it. And then, on my diagram, I have two magnets here, and then I have sort of Romeo and Juliet on this side. They have long-term planning and all kinds of other stuff that they can use to go around social and physical barriers. And in between, you have cells and tissues on autonomous vehicles and Roombas and dogs and everything else. So that's my definition of intelligence, is this kind of some degree of competency to get your goals met under different circumstances. And biology has incredible examples of creative problem solving, where cells and tissues use their genetically encoded affordances, so various mechanisms, to solve problems they've never seen before, either developmentally or evolutionarily.
Oh, that's very interesting, and sort of these, it's becoming, I think, a little bit more. How should I say? The ideas of intelligence are coming more into the limelight with the popularity of AI, trying to sort of define what is intelligence, and it's an important question that'll have to be answered in the near future. If you don't mind.
Well, what one of the just to
Yeah,
say
please.
something about the AI thing, like one of the big problems that I see, and I've been writing a bunch of pieces on this, is that a lot of the discussion about AI is by people who are not familiar with the diverse intelligence literature, and so they're constantly talking about human brains, human
Right.
intelligence, comparison to humans. I mean, this is like, human intelligence is an incredibly narrow and recent type of intelligence, and I think the whole AI debate will be greatly improved by the consideration that there are many very diverse kinds of intelligence that have nothing to do with humans, and and keeping an eye on what aspects of their construction we do and we do not understand. So actually, Nick Rouleau and I just wrote something that's going to be in Nature Machine Intelligence soon. Just a very short piece with a checklist saying, if you want to have opinions about AI intelligence, here are a few questions that you have to be able to answer. It's just designed to directly put people's finger on how much do we not know, about what it is that makes for intelligence in the biological case, and what are you actually saying when you think that some sort of artificial system cannot achieve what proteinaceous hardware can do. Like what's the actual difference? What's the secret sauce?
Now, I just wanted to get back to some of the biology, because I won't forgive myself if I don't ask these questions. In regards to regeneration, something that Planaria are great at, why do you think it is, in the context of these bioelectric circuits, that for the most part humans have had that ability turned off?
Yeah.
Except for some examples like the liver. But
Hmm.
yeah, why do you think that is?
Yeah, it's a great question. I mean, some mammals can kind of do it. So deer regenerate
Mhm.
antlers. So deer antlers grow at a rate of 1.5 centimeters per day of new bone. Can you
Mhm.
imagine that? Bone, vasculature, innervation, skin, right?
Yeah.
So some. I mean, it doesn't have joints. It doesn't have moving joints, but nevertheless, it's a pretty amazing example of regeneration. I don't know why humans in particular have it turned down, but you can sort of, I mean, here's a semi plausible evolutionary story. I have no idea if this is actually right, but it kind of makes sense. Imagine our mammalian ancestors. So it's a little thing. It's kind of like a mouse. It's running around the forest. Somebody bites his leg off. Now, unlike salamander, where you're floating suspended in the water, and you got a low blood pressure and a slow heartbeat, you're a mammal. You're going to try to put your weight on that appendage. You're going to probably bleed out and get infected. So you're never going to get a chance to regenerate any of it. It's what you do need to do is scar and hope that
Hmm.
you survive, right? And that's it. Nothing's going to regenerate when you're grinding it into the forest floor.
Right. Right.
If you don't scar it over immediately, you're gonna bleed out and get infected. So I have a feeling that what's happened is that in dry air, And of course, the other problem is that in dry air, it's very hard to run the injury currents that you need to trigger regeneration,
Right,
the
right, right.
electrical aspect. So I have a feeling that
Hmm.
what's happened is that it just wasn't worth it because it's too hard in the air. Now notice that the one good example we have in mammals, the deer antlers, is something you don't put weight on, right?
Yep.
The one example that we have is sort of carried around on top and doesn't need to be touched by anything. So I think mechanically, microbiologically, it's just not a great thing to invest in if you're a basic mammal. That's my guess. Who knows if that's really what it is? I don't think anybody knows.
Well, that's a plausible theory. I like that. Uh, Another question for you: how do you see these, or has anybody studied how these circuits might work in motile cells? So sperm cells, cells that don't make contact with other cells permanently
Mm.
or for long periods of time, cells of the immune system, perhaps floating through the
Yeah.
bloodstream.
People, so immune cells, some immune cells do make gap junctions with their neighbors. Some migratory cells, like neural crest, make gap junctions with cells as they crawl
As they
along.
roll
So they
through,
make
yeah.
them and as they roll through. Yeah, so some of that happens, but also these migratory cells tend to be loners. They tend to be cells that do things on their own. They're not
Hmm.
part of they're not part of a collective that needs to together work on some grandiose construction project. They
Right.
have a job to do as single cells. So I think it works out fine.
Yeah, it's
But
simpler.
for example, right, for example, if you look at paramecium or any of the things like that are autonomous unicellular animals, they use bioelectrics within the cell membrane. They have waves of depolarization across their surface to synchronize their cilia.
Right.
They use the same circuits, just on a smaller scale.
Uh, Another question that I was sort of floating in my mind as I was delving down the rabbit hole that is your work. 3D bioprinting, right? This is becoming a thing slowly. I mean, it's not necessarily a go-to for medicine just yet. For skin and some cartilage, it is. But how do you think sort of artificially printed cells might be able to recapitulate what actually happens in situ? I guess you would say,
Yeah.
in terms of these circuits. Is it possible to then to print something and then manipulate it in such a way to restore what should be there in terms of the circuits and the memories?
Well, yeah, it's an interesting question. So certainly there's no reason why you can't use the bioelectric interface in vitro. And we have done with Dave Kaplan, we've done some work on human mesenchymal stem cells in culture. We're having now all kinds of organoids and things that we're growing that where you can use that same bioelectric interface to get them to do specific things. I think all of that is totally possible. And I think in some applications, 3D printing of tissue directly is going to be useful, and it'll have some nice applications. Overall, I don't think that's where regenerative medicine needs to go. I think the big picture is exactly away from that idea of micromanaging and telling
Right.
every cell
Yep.
where to be and what to do. I'm much more interested in high level triggers and some of the things that we and others have found where you communicate early on to the cells and you get them on board. You reset their set point. It becomes a you take advantage of their autonomy and their ability to do certain tasks that are where the set points are encoded, for example, bioelectrically, and then you take your hands off the wheel and you let them do their thing. I think that's a much that's going to enable us to have much more complex and integrated outcomes than trying to micromanage the process bottom up.
Right. Why make it more complicated for yourself? And it just occurred to me an analogy would be, for example, you want to ectopically express a protein like insulin to produce it as a pharmaceutical. You wouldn't build a amino acid printer, 3D printer to build this protein. You hijack
Right.
what you can already do in nature via yeast or bacteria and get them to do it for you. Sort of the top down.
Yep.
That's a very interesting approach. Now, what is the state of research in mammalian tissue right now when it comes to the bioelectrome, stem cells, neural tissues, and even the possibility of treating cancer?
Yeah. Okay. Let's see. So for the kinds, I mean, there's been a lot of bioengineering of things like smart bandages that use electrical stimulation. So like Marco Rolandi and folks like that, whom we worked with, you make kinds of the things that can be used in vivo already, but for wound healing and things like that. For real regeneration in mammals, the biggest thing holding back the field is the lack of physiolomic data.
Hmm.
So you can kill cells, fractionate them, and get a nice tally of all the RNAs, the proteins, the DNA, and all of that. Bioelectrics isn't like that. The minute the cell dies, it's gone. It's only available in the living state, and all our sensors to date are optical. So that means that it's nice when we use very transparent model systems like zebrafish and frog and so on. But when you get to mammals, it's very hard to see what's going on in the deep tissue. And if you don't know what the correct bioelectrical pattern is, you have a hard time cracking that bioelectric code so that you can tell the cells what you want them to do. We can apply the things that we've learned from amphibians, and that's one of the things that we're doing, and maybe that'll work if the code is quite universal. But what we really need is the electrome. And this is something that we're working on in collaboration with a company that's spun out of our lab, which is called Morphogenics. So I have to do that disclosure because there is a company that's seeking to do this, and that's the idea. Now, once we have that that base of knowing what the bioelectric pattern is that corresponds to specific health states, then we can use our computational models to say, okay, but the pattern is wrong now. I know what the right pattern is. Here are the channels you need to open and close to get from here to there. That's what the computational model does. And we've used that before for example, in tadpoles to solve some kinds of birth defects and things like that. Then things will really take off. I mean, the other piece of the technology is the stuff that Dave Kaplan's lab and our lab does, is to make these biodomes. So these are wearable bioreactors, and that's a way to deliver the payload, the informational payload, to the cells. So, for example, for a limb amputation, when you wear this thing, and it's got a gel that contains the electroceuticals and so on. So that's where we are now. I mean, we are now working. We've shown leg regeneration and induced leg regeneration in adult frogs, and we are now trying to move it to mice. That'll be the mammalian application.
Now I've heard some discussion. I've heard you discuss a little bit, and I've read about it as well. Sort of a goal of trying to treat cancer tissue, tumors. Now, try to help me understand, as somebody who was a molecular biologist working in a cancer lab, where everything that we did was sort of take a hammer to the genome, take a hammer to the mutated, tumor suppressor oncogenic proteins, genes and proteins. How would you overcome, that morphology of a typical tumor as a result of a messed up gene?
Yeah.
With this approach with the biopharmaceuticals or the bioelectric, I'm trying to understand how it would ultimately solve the problem. Is it a matter of turning the cells back to a healthy state, or is the goal to eliminate these cells forever?
It's not to eliminate the cells. I can tell you a couple of scenarios that we've done that suggest how it might work. First of all, in the birth defects case, we've shown that if you have tadpoles with a mutant Notch gene, so Notch is this really critical neurogenesis gene. If you have a dominant mutation, the brain is just a complete mess. They have no behavior. It's it's very disruptive. So what we've shown is that if you do that, but then superimpose the correct bioelectric pattern, and you do this by simply opening one ion channel, HCN2, and there's our computational model chose that channel as the one that would be able to get the pattern back to normal. The embryos recover normal brain shape, normal gene expression, and normal behavior. Their learning rates go back to the levels of control, so indistinguishable from wild type animals. So that is an example of solving a hardware error in software. They still have the Notch mutation. We haven't done anything with it, but we've overridden the effect that it normally has on the collective of cells by imposing the particular bioelectric pattern that tells you where the edges of the brain need to be in the different regions of the brain and so on. So that's, and I'm not claiming that's going to work in all cases. There will be hardware errors that we can't fix that way. For example, I suspect metabolic diseases such as mutations in various enzymes. I don't see how bioelectrics is going to solve that.
Right.
But these kind of morphogenetic things, I think you can overcome. And so in cancer, so let's just talk about cancer for a moment. I want to tell you two stories. One is that if you we discovered an interesting population of cells where if you mess with their voltage, what happens is that what it turns out that what they're normally doing is keeping all the melanocytes peaceful. If you mess with the voltage of these instructor cells, they stop talking to the melanocytes, and these pigment cells, these melanocytes, go completely crazy and they turn into metastatic melanoma. So they change shape to become these horrible invasive looking things. They dig down into the brain, into the blood vessels, into all the organs, just like melanoma. There's nothing genetically wrong with those animals. They don't have any oncogenes. They haven't been exposed to carcinogens. There are no mutations. There are no DNA damage points. There's nothing wrong with the hardware. What you've done is disrupted communication. You've disrupted communication that tells those cells to work towards specific goals with the rest of the cells. And when you stop doing that, the baseline is this kind of cancer activity. So the first thing is you can cause cancer without anything wrong with the cells themselves. The flip side, which is arguably the more clinically interesting side, is that you can do the opposite. We've shown that if you introduce human oncogenes into tadpoles, so this is like nasty KRAS mutations, P53,
Right.
the dominant negative P53, things like that. You introduce them into cells. The first thing that happens, and this has actually been known for many years, the first thing that happens is the cells depolarize. They depolarize and they electrically disconnect from the rest of the tissue. Now, here's my interpretation of what's going on. Remember what happens in embryonic development. You start with cells that have little tiny cognitive licones, meaning their goals are single cell goals. Their goals about metabolic states, about proliferative states, about transcriptional states at the level of one cell. What happens in development is that these things, and of course evolution, is that these things connect into networks, and these networks have grandiose goals. The network is, for example, build a salamander limb. How do I know it's a goal? Because if you keep cutting it, they will keep rebuilding it, and then they stop when the goal is achieved. Right? That's how we know. So, but they're working on this incredible goal. Now, no individual cell knows what a finger is, or how many fingers you're supposed to have, or anything like that. But the collective absolutely knows, and you know that because you can do perturbative experiments and see how it responds. Right? It'll keep trying to build one specific structure until actually it gives up. If you keep doing it, it'll give up. But anyway, so what you've got here is a collection of cells that are busy maintaining and building certain organs, and then you introduce this mutation that causes the cell to depolarize and disconnect. As soon as it disconnects, what's really happening is that that cognitive licone, the size of the goals it's pursuing, shrinks drastically. Another way to put it, the paper where I first described this is called the computational boundary of the self, because what's happening is the boundary between self and world is shrinking. That cell, that transformed cell, sees the rest of the body as just external environment. Of course, it's going to go where it wants. It's going to eat what it wants. It's going to proliferate as much as it wants. It's an amoeba now, and the rest of the body is just outside world. That boundary between the self and the outside world, the things you actually care about maintaining, which is everything inside of your own boundary, is flexible. It grows during development and it shrinks during cancer. And so these cells are not more selfish than normal cells, which is often assumed in like game theory models and so on, that they're less cooperative, more selfish. I don't think they're more selfish. I think they just have smaller selves. They're exactly as selfish as every other biological. It's just they have tiny little selves now. So that leads to, now that's a very unusual way of thinking about this, right? And it leads to a very straightforward prediction, which we tested into a roadmap of therapeutics, which is, let's not try to kill the cells. Let's not worry about getting rid of the oncoprotein. Let's just forcibly reconnect them to the rest of the network. So when you do this, and there's a number of ways to do this. The cells don't make tumors. They continue the oncoprotein. So I have this image that we show in the papers where the oncoprotein is fluorescently labeled, so it's in red, and you see this huge bolus of oncogene where you injected it, and then it's actually kind of all over the place, and there's no tumor. You look at the same animal in regular normal light, there's no tumor, because the physiology is telling this group of cells to work on nice skin, nice muscle, various other things. So that, I think, is the strategy that we're interested in, is letting the other cells. I don't want to try to tell that cell what to do. I want the other cells to tell it what to do.
Right.
The collective already knows what to do, and so all we need to do is reconnect it. So the status of that is, we showed this in various ways using optogenetics and so on in frog, and we are now in human cells. So we've done some work on glioblastoma. We've done some work on colon cancer spheroids, and that's where we are now, trying to understand how do cells communicate with each other, what are they saying to each other, and what are the electroceuticals that we can use to facilitate this in vivo as a cancer therapeutic.
Dr. Levin, what a fascinating time to be a scientist. As we wrap up, we have 1 or 2 minutes left. If you don't mind, what words of advice or wisdom would you give to students who are looking to pursue this field of biology, which is not as pervasive as your classical molecular biology might be? What would you recommend to students who are interested in this sort of thing?
Yeah. If you remind me later, I'll give you a couple links, because I've written on my blog. I have a couple of posts directly speaking to that issue. The biggest thing is, well, okay, so specifically with respect to bioelectrics, I get a lot of emails about people saying, I want to study bioelectrics. Where can I go to graduate school to study bioelectrics? Right now, there are very few places. There's probably less than half a dozen labs throughout the world where you can actually focus on this. I would say don't worry about any of that. Go learn the conventional paradigm. Go learn cell biology, cancer biology, bioengineering. Learn the paradigm, understand its benefits and limitations, so that you can break it later when you're independent, and you can address the problem in other ways. So I would say learn all the tools that you need for this. Now, to really do this, it's actually a very interdisciplinary field, because understanding computer science, not bioinformatics, but actual, like, real computer, and not not Java coding or anything like that, but actual computer science is very useful. Some aspects of animal behavior, of psychology, of cognitive science, of neuroscience, really useful. So those are the kinds of things. Build your toolkit, and then you can address these problems in your own way when you're independent. That's if you can't find a lab that focuses on this, which hopefully there will be more and more of. My students, other people's students in the field, have gone off and started these labs where you could do this, but still there's not that many of them.
Wonderful. And perhaps in the future we can talk about this at a later time, but you do have a very interesting story of how your family came from Russia to the US, and I heard you say create your own curriculum. If you can't find something that works for you, create your own. Dr. Levin, it was
Yeah.
absolutely fascinating. I will continue to follow your work with great interest. Not only an
Thank you so much.
amazing scientist, but also a great photographer too. Check out his work at his blog, which I'll link down below.
I appreciate that a lot. Thank you so much. Thank you for the conversation.
uh,
Happy to talk anytime.
Absolute pleasure.
All right. Thank you.